Richard, Juhi, and Eugene. Another Eugene. Okay, let's see who has to leave first. Chui, I know you're in Korean time. Do you want to go first?
Yeah, sure. I will share my screen if that's okay.
Yep.
So, our team rewrote the summary and this research panel section, and we wanted to show it to you and discuss with you. So yeah, so basically this is the, can you see my screen?
Yeah.
Okay. This is the summary section that we wrote. So it's the basically the abstract. And I just was wondering if you could give any feedback regarding the abstract section. We just tried to be as succinct as possible, but we still have to cut down 56 more words from the abstract. But yeah, like.
Yeah, I mean the detail. I like the background, the methods such as English, so because the methods will be described more in detail in the actual script, you can put down many, you know, PubMed, Cochrane, Embray, the English text, all these things can be omitted, because they're going to be described in, so if you want to cut down words.
Yeah. Okay. So do you think we should like, I wasn't like 100% sure because like, according to their guidelines, they made it seem like we should talk about some parts of it, but like how much? Huh?
In the abstract?
Yeah, well, like the abstract guideline was like including these. And I wasn't sure like how much we should talk about the ones they want us to talk about and how much we can cut down.
So I think if you have 300 word limit, you can cut down, some of the details, because I think a lot of it more for, trials or, things with more.
Okay.
Yeah. But if you want to cut down, method is better.
Methods.
Yeah.
Okay, sounds good. We will go through that and cut it down. And then that's our findings and the interpretation.
So you emphasize low sensitivity for breast.
Should we take it out? Like, we were a little confused about what kind of results to include here in the abstract. Like should we only include the ones that are meaningful, like that has like clinical significance, or should we because here we didn't include anything about heterogeneity because we couldn't explain heterogeneity through subgroup analysis, which is why we took it out. But like, is it something that needs to be mentioned in the abstract, or is it okay if we like take out parts because we couldn't find anything significant? Like, we're not exactly sure what results should be included in the abstract.
So maybe go down to conclusion in the manuscript. And what were the words used to describe heterogeneity?
You mean the results portion?
Yeah, so
Okay.
where's the heterogeneity portion?
Yes.
Uh, well, so I guess heterogeneity according to, so you go on to talk about stage?
So we did the subgroup analysis and looking at, the different stages, cancer types.
And also for the gender of the population and study type or test type, assay type, but none of them except the type of cancer or
So
stage.
you describe
Like
stage
none
and type
were.
of cancer, but what about the assay platform? People would be interested in knowing, DNA based versus
Yeah.
others.
So as you commented on, we added additional analysis um, on different type of tests. So we classified our test into two groups, only using cfDNA, and then the ones that use other than cfDNA, like we'll call them as multi-omic. But then among 10 studies included in meta-analysis, only 2 were grouped as multi-omic, and then results are not that significant. So it was kind of negative finding in terms of subgroup analysis. And we we added that figure in our figure, but I missed describing that result in the result section. I'll do that.
Oh yeah, that's important. So show me the figure.
Juhi, you can scroll down and then can you go to the figure? Actually, that is supplementary figure 1.
This one?
Oh, below that. Yes. Yeah. Supplementary figure 1. ENF is about, they are about the type of test. We can definitely change the name. Then as of now, I put the name as single and multi-omic for the assay type. It's a bummer that we only have 2 articles using multi-omic tests.
And what were they?
So Cohen and Lennon, they used protein. Lennon used protein other than cfDNA. Then Cohen, Cohen used 8 circulating proteins plus cfDNA mutations, and then for Lennon, they used Oh, they also add some protein biomarkers on top of the gene mutation. And those two, the other 8 studies, they used only cfDNA from gene mutation, like gene.
Mm. What about
The thing,
Lenin?
but the thing is, Lenin is the earlier version of Cancer-seq,
Hmm.
and Cohen is like the actual Cancer-seq.
Okay.
So both of them are Cancer-seq.
Why? Like,
Mm,
so I guess the only difference is here, sensitivity for the Lenin. That just falls really low.
um,
Maybe the
yeah,
sample size is
Lennon,
really small. Why is that?
um,
Well, I guess the coin.
yeah, weirdly,
Oh, I guess the.
like, yeah,
Oh, I see. The coin is case control and Lennon is perspective, right?
Lennon is cohort
Yeah.
Study. Yeah.
study.
So you have very few events.
Yeah, probably, yeah, totally.
So, I mean, it's going to be very difficult to claim that multi-omics for sensitivity had inferior outcome because they're so different cohorts and
Statistically, strictly speaking, there's no difference. So it looks like there's a difference, but then if you see the result, like confidence intervals are overlapping a bit.
Mhm.
So I don't think we can argue there's a significant difference.
So I think I would at least add in the results that whether it's blood circulating DNA based or combination of proteomics. It's not even proteomics, it's multi-omic assay versus single circulating DNA based assay. There was no difference between in sensitivity and specificity. So I would emphasize that. Because people, I mean, we did the analysis, so.
You mean in the results section or the
Yeah, in the
Okay.
results section and in the abstract.
in the abstract too?
Yeah. And you can kind of merge that. You said there's no difference in stage, there's difference. So subtype, there's a difference. Well, I guess there's no difference for, according to the assay platform or, make it succinct. And there was no difference in gender too. What else did you look at? Eugene?
Gender, study,
Huh?
uh, yeah,
Gender and
gender,
study type?
definitely we looked at. Yes, we try to classify studies into cohort and case control,
Yeah.
and then there was
You can do study
other
method,
than as a type.
study method, and open parenthesis, case control versus cohort study. So there was no difference.
Okay, we will clarify that. Yes.
So you could,
Types or
yeah, mention all
there
that
was-
in the abstract results section,
Okay.
all in a
Right,
lump.
yeah.
Yeah.
And the current results in the abstract, like, do you see anything that can be deleted or modified?
Uh, looks okay.
Okay, so we keep whatever is there right now and we add the subgroup analysis.
Yes.
Okay,
You don't have to put any number to it to say there was no difference, no.
okay,
Got it.
So interpretation, you have ownership, responsibility, economic value. I don't know, that's kind of not, it's not something that's driven from the findings, the ownership, cost effectiveness. So you could kind of make that, you know, the however and nevertheless. Something like very more succinct, like although there are issues related to, you know, practical application in clinic, you know, something like make it very vague. There's going to be multiple barriers. Because these all like details will be discussed in part in discussion,
yeah,
not, it's not real interpretation of the finding, right? So yeah, I would just cut that down more as a barrier to practical application or something.
okay. So are we talking about the however sentence or both the however and the nevertheless sentence?
So however, to nevertheless, just make that, you know,
Okay, okay, I understand.
yeah. Just make that into one phrase.
Okay,
Although these things, practical issues exist, MC
Mhm.
test may, you know, something like that.
okay, sounds good. And then they have this separate portion that they want that is different from the abstract.
Okay.
It's called research and context.
Mm,
So it's like a panel
mm,
and they want to see like evidence before this review paper.
mm,
And then they want to see added value and implications.
mhm.
So we tried to draft it out and was wondering what you thought about it.
Hmm. So I don't know if NCCN is the right,
Okay.
NCCN is more for people with cancer diagnosis, what therapy to give. So
Mhm.
the most widely accepted is US screening task force.
US Screening Task Force.
Uh-huh.
Okay.
You can Google the exact. I mean,
I
that's
think
the
I saw them. I'll Google
yeah.
it again.
They're the one who like recommends, you know, annual CT for smokers and, you know,
Oh, okay.
yeah.
Okay. Okay.
So when you say campus cisternos, is that
Oh, this is just citation, sorry. These are the
Okay.
review papers that deal
Okay,
with cell-free DNAs.
So you're gonna change that in citation.
Yes.
Okay, good.
Okay. And should we include anything else here?
No,
Okay. And the added value.
so added value, you put the search of the data. But that feels like method.
Mm,
But does that talk about method?
I will double check, but the guideline was, I'll double check.
Hmm.
The guideline made it feel like I should talk about method, but we'll read through it again.
And then you have implications.
I wasn't sure if placing that statement there is correct. The assuming an annual.
So explain how you got this point.
So I got that point from one of the articles that we included. Basically, like in the conclusion, they were talking about how like their screening tests have like a PPV of, I think it was like 51%. And that given that like the bare minimum required for screening tests of single cancers is like around 4% for different cancers, as depending on the US Preventative Task Force. The tests that we analyzed were like had PPVs of like much greater than 4%. And so like I wanted to support the claim that Juhi made that our test had high diagnostic accuracy. I don't know if like that's like the correct way to do it, but that was like in my mind like a way to like quantitatively say that they have like a high accuracy.
Hmm. What was our PPV number?
Actually, for us, we were not able to calculate PPV. We didn't pull PPV because PPV is affected by the prevalence, and most of our meta studies were done as case control, which applying like prevalence doesn't make sense. But I think instead of PPV, we can argue about the AUC actually under area under curve to support this kind of a sentence, this kind of statement, because AUC was for us for Meta, it was over 8 point, it was about 87%,
Hmm.
which is generally considered to have excellent accuracy for diagnostic tests. So yeah, if that's okay, maybe we can argue about, like, we can introduce our AUC rather than PPV. But then, for I think, yeah, I think I know where a researcher is coming from, because most papers,
Hmm,
whether they are included in our meta or not,
hmm.
some papers I noticed they just presented their own PPV based on their just their research, and then apparently those PPVs seem to be very high, relatively high. So although we cannot pull them up, maybe we can just describe. Maybe we can be descriptive too.
So for, I know we can't do that for case control, but for cohort studies, are we able to report those for cohort studies?
Uh, yeah, because we have our because we have wait, yeah, PPV for only cohort studies. I think so. We didn't do that as of now. But then, do you think we have to calculate like NPV as well, or are we good to have PPV only, like positive predictive value and like negative predictive value?
I think you have to do both.
Yeah, if we have to do one, maybe.
So how many cohort studies do we have?
Among 10 included in meta, cohort only 3.
Only 3 cohort studies?
Yes.
Hmm.
Oh, sorry, 4. We at the very last time we had 1, so it's
Or
in total 4 cohorts.
no, to number 1, I would mention AUC. Number 2, I would do positive predictive value, negative predictive value for cohort studies, make similar tables. And then we cite our own PPV from our meta-analysis, cite that number and say it is better, greater than the USPF. This is the US, is a what, preventive
force.
task force. Yeah. So yeah. So let's do that. Just one more analysis.
Okay.
I also wanted to clarify, so can we take this sentence out and put it in the added value instead of implications? Or is
I think
implication,
it added value. Yeah.
okay. Is the rest of it okay?
It's okay.
Okay. So another thing for the introduction section, Richard was like Richard found this cool thing Biden said. President Biden apparently like made an announcement about Moonshoot program, which is basically early cancer screening, multi-cancer screening, using a blood test. So there was this quote that he said, imagine a simple blood test during an annual physical that could detect cancer early. And we wanted to run by you to see if you think it would be nice for us to include this in the introduction or the discussion, or if it's too casual, so it's better without.
I think it's better without. It's too casual. It reads like newspaper.
Okay.
Good, nice try, but we can-
We tried.
you know, Biden came to AACR and gave a speech.
Oh,
Biden's
really?
son is
Is
also
that where he got the idea?
yeah, you know, Biden cares for cancer research, and Biden
Wow.
his son, who's a very accomplished politician himself, died of glioblastoma.
Oh,
So he is a big proponent of cancer research. Yeah.
wow. Yeah.
You know,
Um,
Biden's pretty old, so his son is pretty old too. You know,
oh,
it's not like son died young. We're a very accomplished guy.
mhm.
But anyway, go ahead.
Um, Eugene, did you have some stuff you wanted to run by? So these are the changes that we made. We will change them as per your feed, as per what you said today, and then we will re-show it to you next week. But one second.
Sorry, my heard.
Was there any additional things that we wanted to talk to Professor Che about?
Dr. Shay, do you think it's the right time to show you all the figures and figure legend
Is
as
it
of now? Or
ready?
oh, but then we have to add like PPV and MPV. So would
Yeah.
it be better to show you next
No,
time?
no, no. The rest of it you can show me now.
Okay, then Juhi, could you go to the very end of the
Which
menu?
figure?
So from figure
3?
3, yeah,
Or-
from figure 3,
Okay,
yeah.
yeah, I'm at figure 3. Is this good, or do you want me to zoom in?
Zoom in a bit. Yeah, it will depend on each figure, but for this, yeah. Zoom out, sorry. Yeah.
Oh, zoom out. Oh, sorry.
So Dr. Say, this is our main kind of the general forest plot for sensitivity and specificity, and including 10 studies for
Mm.
meta-analysis. And then I'm not sure whether legend is enough as of now. Juhi, could you scroll down a bit?
Yeah.
Mm. Yeah, I think it's okay.
Okay, for the next figure 4. Figure
Mm.
4 is summary receiver operating curve. And I try to kind of explain about each component, not explaining the meaning of it, but
Mm.
yeah.
Oops. Sorry, I changed it.
So SROC, summary receiver operating curve.
I noticed many like meta-analysis about diagnostic tests, they present this kind of a figure.
Mm.
But the only meaningful thing I noticed is AUC, so that I really want to interpret this figure more, like maybe what kind of that 95% CR region mean, and but then I was not able to do that up until now. And each like triangle signifies each test, each study that isn't bettering. that is included.
Yeah, it looks okay.
And for figure 5, it's a little com compact. I'm sorry. So it's kind of a main result of our meta. So stratified by cancer stage and type. So A and B is about they are about stage, and then C is about cancer type. But then because we classify the stage into like in two ways, one is 1, 2, 3, 4, and then the other is we group 1, 2, and 3, 4. So I try to present all that result in at once. So does it look okay or?
So is this one figure panel, one big figure?
One big figure. I made it as a one file, and then for the resolution, I don't think we have to be worried about it, because when we actually submit our article, maybe I can make a high resolution figure about of this and then submit it.
Okay. So yeah, just work on the ABCs. They kind of,
Okay,
so
they don't look
ABC has to be,
a little bit more smaller. Right?
yeah, smaller, not overlapping.
Okay,
Mm.
and then legend is pretty same with the previous one.
No, this is what I would suggest. Go back up. Figure 4, go back up. Figure 3. So I want you to do the same for this ABC.
Mhm.
It can be even longer. I know it's going to be really long, but
Oh,
how do I describe this?
so you want me to put all figures in a row, not like in parallel?
Mhm.
Mm.
And when you upload, so at least I want you to have the same size of each ABC for figure 5 as well. Now you're gonna run into issue of it being too long. So it's the journal staff to decide how do you want to assign it,
Mm.
but at least the you know, they should be able to look at it with at least this size.
Okay.
So you can have it's figure 3A, B, C. So you can have it as one file for each. You don't have to have like one big panel. You can have figure 3A, figure 3B as a separate figure file. You understand?
Oh,
It
I
doesn't
didn't know
have to
that.
be one file.
Okay. Oh, really? Oh, yeah. Okay.
No,
Based
no,
on my experience,
what I'm
yeah.
saying, what I'm saying is you can for the review paper,
Mm-hmm.
listen hard. Review paper, each panel could be one file, picture file, so that the reviewers can look at it with high resolution.
Mm-hmm.
Now, with the same resolution, you can make that as a panel when you actually upload, because when you upload in the journal website,
Mm-hmm.
sometimes it will ask like one figure is one panel,
Yes,
figure one
yes.
ABC. So when you upload it, you can make a panel for uploading purpose only.
Oh, okay. But then I can
For
put.
journal reviewer, you can make one file for each figure A, figure B, AB, 3B, 3C, 5A.
Oh,
So that way it will still stick, you know, have a high resolution for the reviewers.
as a format of document, I can put
Yeah,
all
yeah.
of them in. Yeah. Okay. Got it. Yeah. Okay. Yeah, I can do that. I already have each file as a separate file. So yeah. Yeah, I would do that. And then for figure 6, if we can move on,
Mhm.
it's about accuracy of tissue of origin localization.
Mhm.
And yeah, it also
Yeah,
has two
same thing.
components.
Yeah, same thing, make it bigger to
Okay.
read.
Yeah. Oh, okay. And for yeah, but then no, the figure 6 has doesn't, and then
Why is this red?
you want me to be consistent? I use a different, so
It doesn't have to be colored, so I would,
okay.
right here, it doesn't have to be colored, so I would use the same thing you did with the previous figure.
Okay. Mhm. Okay, I will change the color. And for table 2, so thank you for making notes. And you scroll down a bit. As the gender population test type, I would just make it as of now, but then I can change it as type and stage. In the Oh, and one quick question. I was looked out a bit earlier. Dr. J, do you want us to keep the number as having, under 2 points or
1,
3 points? Like the digit below the 0, like point?
so here it's,
Do you want us to round up
It's a-
or?
so when you say sensitivity, we don't use percentage? Is it?
We use, yeah, we use percentage, but then if you do meta, they just give us results with not percentage but just proportion, because sensitivity basically it's proportion, right? It's true positive over true positive plus first negative. So I'm not sure whether we have to use proportion or percentage, but for the meta result they gave us just proportion, not percent.
What about the heterogeneity? Do you use percentage there?
Yeah, heterogeneity usually it's presented as percentage usually. Yeah.
So explain the sensitivity, specificity, and heterogeneity and test for subgroup differences. But how is that sensitivity, specificity different from the first two columns?
Oh yeah, so yeah, it to me even to me it looks a little bit confusing, but the reason I put sense and spec in the column called heterogeneity is that I just want to report that there's a heterogeneity for each of the results. So it's a little bit hard to figure out what would be the best way to present all these results. So maybe I can do like, I can put heterogeneity right before the sensitivity and specificity column. If I make each heterogeneity column for each of sensitivity and specificity, I can avoid just put
Yeah,
two words here. Okay.
yeah, that's better because it's hard. So for sensitivity, and then you add heterogeneity and test for subgroup of difference.
Okay, so like this, maybe. Mm, yeah, maybe I can. But then like, yeah, okay.
And you say heterogeneity for sensitivity or something, make sure that's it.
Yes, okay. And for the subgroup differences, chi-square value, I would do the same thing, maybe.
Or one way is for the second, third column, you could put like sensitivity as a box that covers both columns up at the
Okay.
top.
Mm-hmm.
Yeah,
Oh,
like sensitivity. Yeah,
Yeah, and then I can just put confidence interval here.
yeah. Well, sensitivity and then leave it, and then you can get for sensitivity out, and then
Yeah,
yeah.
it looks better. Yeah, thank you for your input.
yeah. So you can take out for sensitivity in front of after heterogeneity. Yeah. Yeah. This is much better to understand. Uh,
Okay.
now I wanna. The p-value is okay, 2 digits after decimal point, but
Because in the figure we have 3 digits, so I just follow that rather rounding up, but
yeah, let's leave it there. Yeah, it's okay.
okay.
I mean, other papers are, maybe doing the same thing, right?
Yeah, but I will double check. Yeah.
Yeah.
Okay, almost there. So for supplementary figure, so basically it's kind of redundant. We here in the table as a form of table, we already have all the results about subgroup analysis. So I think that's why you suggested us to have actual forest plot as a supplementary figure. So I made that as supplementary figure 1, but it only has results from study type, gender of population, and then type of essay, test type. Yeah, I would be consistent for the names, but then still. And then for, do you want me to put all these figures, 6 figures in a row as well? Like just for the purpose for reviewing, like for reviewer to
Because
see
this is
clearly?
supplemental, it's okay.
Okay. And we have last supplementary figure 2. It's about specificity, not sensitivity. We have sensitivity results as a main figure, but then for specificity, as I explained before, yeah,
Yeah, it looks okay.
okay, okay, great. So we will modify some figures or tables as you commented, and then yeah. So that's all for me.
So that's what we have for now. We'll try to revise everything. And then show it again next week.
Okay,
Okay. Sounds good.
good job. I mean, let's get it wrapped up soon because
No.
it's been a while and
We
I'm
really
afraid,
try to do that. Yeah, but
uh,
then,
I feel bad for everybody because we're meeting like
yeah,
twice a week. But yeah, it's almost
Yeah.
there.
all right. Anything else to add? No? Okay. Okay, Richard, do you have anything else? Besides this?
No, nothing else to add. I'm just
Okay,
getting a vibe.
okay. And for the DART paper, you're working with a statistician?
Yeah, I'm like waiting for them to reply. They haven't been like very responsive, so I'm trying to like push them more. But
Do they ever respond? No?
they responded one time. They were saying like we're working on it, and then that was it.
Okay, I would suggest when you email that sometimes there's a, contact, and I would just give them a call. Or there's a room number, I would just visit them and like, hey, I just want to put, face to a name, and then I'm Richard, and yeah,
Yeah, I could do that. Yeah.
you have to be persistent. These people, like, they get paid the same way. They don't have to do this. You know what I mean? They're like, it's just let's put it this way. Their boss is not pushing them to do this project, and there's other priorities. So
Yeah.
that we're not paying them directly. I mean, it's coming from Cancer Center Fund, but it's their job, but then we have to just gently push them.
Hmm. Yeah.
Oh yeah, I'll leave it up to you. This is all called social skill. Get things done. How do you get things done?
Okay. Okay. Yeah. I'll try to figure it out.
Yeah, it's not, yeah, but all right, good. So nothing else, Richard? Just before you get it, just write introduction and discussion about like what does the literature say about this cohort, and look at other DART paper and just start writing it. Okay.
Yeah. The intro is done. I think I'll wait for the like results before sending you like the discussion and everything, but part of it.
Yeah.
It's good. Yeah.
Okay, good. We have Minjung in Korean time too. Minjung,
I'll turn
let's
my,
update.
I don't have I only have updates regarding the interferon gamma pathway. But honestly, I think the more I study this, the more I'm getting lost. So I'll just show.
That's okay. That's why we're discussing. Hey, Minjung, why don't you do this? How about you invite Dr. Taegon Kim to our next lab meeting? Can you send Taegon Kim like invitation
Mhm.
anytime between 8 to 11, if you can spare maybe like 20 minutes, 15 to 20 minutes of your time discussing your own paper and the pathway that we have questions for you. And then coming from you, you can CC me, or maybe it's best to do that to the, because he's on the messenger, or I don't know if you know his KakaoTalk. You know what's the contact number that you know?
Messenger.
Messenger? Yeah, he uses Messenger. So just Messenger, text him. You can put me in the same room.
Oh, okay.
Let's see if we can invite him for our discussion.
I'll do that.
for next week. Yeah. But let me know what you found now.
Uh, uh, yeah, I think it might be better if I present next week when Dr. Taekwon Kim is in the lab meeting, because I am getting, yeah, I'm not sure if I have a grip on the role of interferon gamma in the tumor microenvironment. But I guess just to summarize what I've been looking at in the
Yeah,
past
yeah.
week. So last week we left off with, I think the main question being, what properties of interferon gamma signaling may contribute to adaptive resistance,
Mhm.
and particularly what are some of the spatial temporal characteristics of the signaling.
Mhm.
And oh wait, I should.
Yeah. Good.
So this was the paper that I briefly showed last week,
Mhm.
particularly on what happens with prolonged interferon gamma signaling. And this was the figure that you summarized, like impromptu last week
Yeah.
at lab meeting,
Yeah.
where when you have chronic interferon gamma exposure, then it basically leads to the expression of inhibitory receptor ligands here, including LAG3, TIM3, LAG, TIGIT, etc., some of which were equally expressed in some of the hyperprogressive disease manuscripts that we looked at in our meta-analysis. So that's where I hypothesized that interferon gamma signaling may be related to T cell exhaustion. And in this paper, when they knocked out JAK, or when they basically blocked the interferon gamma receptors, they were able to, the exhausted T cells were able to recover its previous state, and it was able to make an anti-tumor response. And this was a paper from 2016. And this same group at UPenn with Dr. Min, they published another paper in 2019, which was a more comprehensive picture of what is happening. And that is this paper here. Opposing functions of interferon coordinate adaptive and innate immune responses to cancer immune checkpoint blockade. And so to summarize it briefly, the top portion here describes the tumor interferon gamma driven resistance. And when they characterized the ISGs, which represent interferon gamma stimulated genes, they realized that in patients who have resistance to ICB therapy, the immune cells are characterized by a decreased expression of these ISGs, whereas the cancer cells are characterized by an increased expression of ISGs. And what they did was they realized that even with the same interferon gamma signaling, the expression of associated genes is differentially expressed, depending on whether it's a tumor cell or if it's an immune type cell. And so what they did was they blocked, they basically knocked down the interferon gamma receptor in the tumor cell specifically. And what they found was that by knocking this receptor in the tumor cell, you would expect the ISG expression in the cancer cells to decrease, right?
Right.
A finding that was novel in their paper was that when this ISG expression was decreased in the cancer cells, conversely, ISG expression, which was depressed in the immune cells previously, was increased. And as a result, in the case that the tumor microenvironment had adequate neoantigens or MHC1 expression, then these exhausted T cells were able to mount an attack using a mechanism that was primarily based on perforin granzyme B activity.
Mhm.
And in the case of tumor microenvironments with poor neoantigens or MHC1 expression,
Mhm.
they were able to regain anti-tumor response, not via perforin granzyme B activity of T cells, but instead through the maturation of NK cells.
Mm.
And so I thought this was an interesting update on what prolonged interferon gamma signaling does to different immune components of the tumor microenvironment.
Mm. So let's
and-
stop right there. So they just added, is it a monoclonal antibody for interferon gamma receptor of tumor cell? Is that how they
So
block?
they basically knocked it out in their mice models.
How it knocked it out? Okay.
Mhm. So that's how they
They
did
could
it.
selectively do that for just the tumor cell in the mice. Or is that mouse model meaning like all the, well, I guess they knocked it out in tumor and then
Right,
injected that into mice.
right. So they
That-
usually graft it onto the mice.
But that tumor
Mm.
had this gene knocked out,
Right.
interferon gamma receptor gene. Okay.
Mhm.
And then they looked at the two different type of immune cell. One is the terminal CD8 T cell effector. And then there had adequate neoantigen and MHC1.
Mhm.
So the tumor had the adequate neoantigen
So
on
what?
MHC1, right? So how did they control this adequate and poor neoantigen?
So they basically had a cohort of patients
Mm-hmm.
and they characterized their immune contexture and expressions of neoantigens, MHC, and so on. And I think they divided the cohort.
And so they had two different a tumor grafted in different mouse cohort. Okay.
Mhm, mhm.
So that's the two different. So with the good neoantigen and good MHC, good antigen, it just basically means very good antigen presentation made so that the effector T cell recognizes and kills through the perforin granzyme B.
Mhm, mhm.
If you don't have that good antigen presentation, then you're gonna have, immune cells are still like increased in ISGs, but then there's interferon gamma will not work through this antigen interaction presentation, but it would go through more innate. So the macrophage natural killer cells will be activated, right? And it will have trail receptor is another like killing pathway, right?
Mhm.
So in any case, interferon gamma in the blood, I guess, can initiate adaptive or innate immune response killing cancer. But in order to do that, I guess the chronic stimulation of interferon produced by tumor cell has to go down. It's like paradoxical, right? Interferon gamma comes out from cancer,
Mhm.
or the interferon, or is it the receptor? Where they block the receptor, interferon gamma receptor.
Mhm.
I guess interferon gamma receptor will turn on the JAK2 STAT1, and that pathway creates PD-L1, correct?
Correct.
So if you block that, then you won't have PD-1, PD-L1 on the surface. Well, PD-L1 on the surface.
Mm.
Maybe with other checkpoints as well, but in any case, if you block it, then the tumor's adaptive response to avoid immune response goes down. So that I think that's what they're trying to show.
Mm.
Um, so interferon gamma will, if presence, will exhaust T cell in the blood. But that exhaustion usually helps, happens through the immune checkpoints, which are induced by the tumor surface interferon gamma receptor.
Right.
Yeah.
So that's what they consider the paradoxical effects of interferon gamma, in that it's helpful for the immune cells at first, but later it becomes helpful for the tumor cell to evade basically immune response, because it induces later on the expression of PD-L1, which ends up exhausting the T cells.
So that's why we have, so you see that even without the effector T cell, the innate, immunity can still kill cancer cells. That's why we have a lot of innate immune response immune therapies now developed. There's, so go on.
So that the differential expressions of interferon gamma stimulated genes, I thought was an interesting point to consider. And secondly, I wanted to find out whether there are any studies out there on how interferon gamma is distributed within the tumor microenvironment. And this was a paper published 2 years ago. And it talks about how basically interferon gamma activity within the body usually takes maybe, so sorry, let me rephrase what I'm going to say. So interferon gamma production happens through a course of only 2 to 3 hours. But the reason why interferon gamma is allowed to make such a prolonged effect is that it is able to diffuse throughout the tumor microenvironment instead of being just focused at the, here, it's hard for me to describe. Let's see. Maybe this can you see the mouse cursor as well or no?
Yeah, I do.
Yeah, I think maybe this might help. So this is from an immunology review paper on interferon gamma that was published March of this year. And what I found to be interesting was that the way interferon gamma is basically released from each type of immune cell is different. So if you look at the NK cell here, it basically, there is no sense of direction in interferon gamma release. And when you look at the T helper 1 cell, it's very narrowly directed within the immune synapse. But what's interesting about cytotoxic lymphocytes is that it's called a leaky synapse. So most of it, usually perforin and TNF, are directed to the tumor cell, but interferon gamma, interestingly, has some room to diffuse elsewhere.
Hmm.
And with that in mind, what they found out about the spatial temporal activity is that the interferon gamma that is released from the tumor site is able to diffuse to a distance that was further than expected
Mm-hmm.
and induce the same type of interferon gamma stimulated gene expression activity. Does that make sense?
Where?
Throughout the tumor microenvironment. So whereas other cytokines may have more limited spatial distributions, interferon gamma can spread far more widely.
So I guess that's like T cell activating each other. You can activate innate. So large field. So I mean it says modified tumor microenvironment, but I guess it's to excite few other colleague T cells nearby. Or maturate them.
Maybe I should. So let's see. Actually, no, I don't think I can describe the results confidently, but
Why don't you send, well, I have two ideas. Send these articles
Mhm.
Taegun Kim and invite Dr. Daniel Shin as well. It's his area of expertise too. Melanoma interferon gamma pathway is
Mhm.
where it's his PhD. He did PhD with Tony Ribas. So that's his expertise too. But I don't know if he can come in the morning, but maybe he can. It's LA time, but he can come in maybe like 10, 11 ish. So let's see if both can come in, good. If both cannot come in the same time, we can still ask that question. So
Mm.
yeah, invite Daniel Shin. But well, I guess paradoxical, like chronic stimulation will lead. I mean, we see that in biology, right? In physiology, what is that called? Like the calcium voltage, gated channel. If you have stimulus over and over again, then paradoxically it turns, into a more like a negative feedback. And it's like a fatigue. What is that called? That phenomenon in physiology. You guys remember? Repetitive stimulation leads to inhibition or fatigue. There was a terminology.
Desensitization?
What did you say?
Desensitization or like exhaustion?
Something like that. But I thought there was a word,
Not sure.
right?
Oh.
But if anyone, can think of it later, let me know. But we'll move on. But Minjung, anything else?
No, I think I need more time to study.
Yeah, no,
I'm,
this is really good question. We keep reading, find relevant. Maybe there are papers that aren't published or there are published that we didn't find yet that could maybe explain this better, but go in deeper into more gastric immune microenvironment. You know, why do they have higher hyperprogression rate?
mhm.
And the anecdotal evidence, including my case with medullary thyroid cancer, is that for those ones that looks like, so this is an interferon gamma. So the gastric is not even MSI high. When we say hyperprogression in gastric, that's not the MSI high gastric, correct, Minjung?
It wasn't.
It wasn't, right?
Mm-mm.
Yeah. MSI high tumors, I don't think they usually have higher hyperprogression rate. Usually most of them respond very well. But it's usually the cold tumors. Like a lot of gastric tumors are mucinous. So if I give immunotherapy to mucinous, let's say lung cancer, I know Dr. Jian is looking at transplant for, but this mucinous type, what's so special about it? So maybe just the cold tumor, the TIL, and why is there interferon in gastric non-MSI high? You know, what does it do? We thought it was cold tumor. Maybe it's not. Maybe it's T cells are exhausted already, but it's there. And with immune suppression, the cancer can grow faster. So the correlation between interferon gamma type and cold tumor and borderline tumor, marginal tumor, you know, what is the relationship there? Let's keep reading about it, Minjung. I think we're close. This is the time to create theory of our own and see, like I said, TCGA. You know, remember I said maybe you with Eugene O, look at the TCGA and look at what kind of subtype. So so I said maybe Jian and Jisan can learn from this TCGA. We know Wangyong cannot come. Wangyong might be able to do research distantly. I don't know. She still have time to do research. So you can still work with Wangyong about the TCGA. She's done TCGA research before in ovarian. But anyway, whether it's ovarian, endometrial, or gastric, but I think gastric comes out because of the hyperprogression rate. What is so unique? And is that interferon gamma type dominant type in non-MSI high gastric? Can you prove that? Because if you go to the immunity paper, they should share us how they classified it. It has to be open source. If not, we can ask the authors to share their algorithm of assigning. And then we can do that in gastric and then kind of see if the hyperprogression correlates. And then we have survival in gastric. So if you have poor immune profile, that might, even without checkpoint inhibitor therapy or relevant therapy, you could portend poor prognosis in that subtype too. So let's explore gastric TCGA and apply the cancer landscape proposed by the immunity paper. I think that will be a nice way to indirectly validate our hypothesis. But again, we need to read more. But I guess, I mean, up till now, it's the chronic exposure leading to expression of negative checkpoints. But if you go back to our very first few papers that we cited about how macrophages play a role in hyperprogression, Then you can read about what does interferon do about, you know, M1, M2 type macrophage. And through that, there could be, we can explain hyperprogression too. So if you know, keep reading, Minjung, I think we're almost there. And then we can invite two of our mentors, Dr. Daniel Shin and Taegon Kim, to give us comment, and maybe they have another idea because they know what literature's out there and what's relevant. So picking their brain, I think, is our next step. Yeah, no, I think this is a great area. And then this links to mucinous. There's a lot of mucinous in gastric. So why is giving immunotherapy dangerous in gastric? We're not talking about MSI high. You know, these Garden people work with Samsung Medical Center, and they publish a lot in MSI high gastric. And then they generalize it to gastric cancer, which I object to. They're very two different groups. But if you give immunotherapy to just the gastric cancer without chemo, it's dangerous. And we have to explain why it's dangerous. So the immunotherapy only trial should be avoided in certain type of cancers. So that's sort of our theme here. Don't blindly give it. And then if you have a biomarker based on RNA transcriptomics that will say, hey, for this type, don't give immunotherapy only. There's a high chance that you'll have actually hyperprogression. So that's where we want to get to, but we want to build a solid biological basis for this. So this is actually the fun part. This is the labor intensive part is the, you know, like the plot, making the plots and all that, but really coming up with theory, this is the best part of science. So keep going, Minjung. We'll do this together as a team. So good job in reading a few more articles, but let's keep doing it next week. Okay. All right. Jian. Let's go to Jian. All right, Minjung, I know it's late there. I'll talk to you later. You don't have to stay. So. All right. Jian, updates?
Yes. I'm not sure my screen. Okay, can you see my screen? So first project is double lung transplantation, a review with the pooled analysis. And I asked to the UNOS, United Network for Organ Sharing database. And okay, the one of the guy, the name is Dennis Tripp. He told me that I can't merge any data with other original data. But anyway, I got the statement yesterday, and so I'm gonna show you. I don't want to miss anyone, but I'm not good at English, so please take a look together. Okay, this is the one. Actually, number one is just a basic principle, I guess. And I think we have to see the number 5. So another attempt in order to permit others to attempt to combine or link the data with the patient level reports in another database or source of information.
Oh, so this can be interpreted as merge?
Well, I'm not sure, but does that mean we can't upload it to any other link or the other place? But I think, does that mean that we can't merge another data with data, even hours.
So combine or link data with patient level records in another database. What do you think, Liam? Is that saying we can't merge two data?
Yeah, it seems so.
Yeah, I think so. So
And
we can I don't know, we can, because it's still mixed with the single lung and then double lung, we can just get the double lung and just show what the double lung data shows, and we don't have to
Mhm.
mix it with other data. We can just present that.
okay.
Yeah, unfortunate.
Anyway, try to request if I get it.
Yeah.
Okay, I will. And there's another principle, number 10. All publication using data will contain the disclaimer in the
Yeah. So we'll add that in our
session.
paper. Yeah.
Okay, all right. Then I will make it. So let's go back to my okay. So I will complete the statement for request and data. And now Jay is review and study about the test, and he's gonna
Mhm.
do journal review maybe today.
Yep.
And Sam is working with the figures about the history of the double lung transplantation.
Right.
And I gave him 2 weeks. Now it's maybe 10 days left. I will check it out.
Good.
And last one, yesterday, Dr. Chen told me that the lymph node metastasis could be one of the factors, maybe the failure of a lung transplantation. So,
So just for
I-
you guys, after Albert Curie, there was one
Mhm.
more patient who got double lung, was successful, doing fine. The third one is waiting for lung, just like Albert Curie. He's intubated now, ECMO, just waiting for lung. So
Mhm.
let's pray that he will get the new lungs.
yeah, I will.
Yeah, just follow him daily with, in the chart.
Mhm.
You guys know who he is, so.
So here is one of the articles that why the lymphatic vessels are so important in the transplantation. So if the cancer has already lymph node metastasis or the tumor emboli, which is filled with the lymphatic vessels, it could be very negative factor for lung transplantation. So I will a little bit more review and then put them in the manuscript.
So why is it so important? I mean, it says crucial. Yeah.
Yes, I have to read because I just found this article. So I will read and I will share the knowledge next to them.
I think I think this is because most lung transplant happen in non-cancer patients. So this is in terms of avoiding graft versus the graft rejection. Yeah. But anyway, so here's the thing. So I think Eugene knows this, maybe, and then maybe Minjung knows this well too. If you can reject organ, you can reject cancer. So, so rejecting organ and rejecting cancer are like similar phenomena. So if you have, so here's the thing, like if you have a lot of transfusion, then you increase tolerance. So your body's now used to a lot of different foreign things. So people with a lot of transfusion history, they have less organ rejection after transplant. Is that true? Eugene, no.
Not that I know of. I've never read that evidence somewhere, but I might. Yeah, we'll try to find that. It sounds like reasonable guess, but
I think maybe I saw that somewhere. I thought you knew, but anyway. But people whose body has never been exposed to foreign antigen, they get transplant, they have higher rate of organ rejection. Okay.
Yeah.
So that is why people with more transfusion
Mhm.
have higher rate of cancer. Because if the immune surveillance system is active,
Mhm.
anything foreign, reject.
Mhm.
Then the cancers foreign, they reject. Okay. So it's a better, well-functioning immune surveillance system. So that goes all, along with the transfusion data. More transfusion you have had in the past, they're higher cancer rate. Because your body immune system becomes more tolerant of anything foreign, including cancer.
Yeah, I got.
Yeah.
Yeah,
So
yeah.
I just want you to understand this biology, tolerance and rejection. But in order for our body to control cancer, we need good rejection.
Okay.
Yeah,
Pitch, I have a question.
yeah.
So I get the rejection concept in immune surveillance, but
Mhm.
isn't cancer technically like self?
It is self,
Tissue?
but it has mutations. So let's say HER2 mutation. The body recognizes it as a foreign peptide antigen. So that is why our T cells are able to attack. Yeah, it's because of mutation that, and then if it's a fusion, it's even a stronger antigen. So it's any peptide that our body has not seen. But our body has generated, the cancer cells have generated those. So, yeah. Anyway, Jiyeon and Jisang, I want you to keep thinking about this, tolerance and
Mhm.
rejection. And this is well described in Eugene Oh's paper, actually. Eugene Oh and Seokju, this
Yeah,
rejection.
Yeah, transfusion manuscript.
yes, Dr. Sen, you were correct in that. For kidney transplantation patients, when people got blood transfusion before getting new kidney, the graft rejection proportion decreased.
Yeah.
Oh.
For kidney transplantation, that's the only evidence.
Mm-hmm. So it's a universal phenomena. Anyway, that's why transfusion is never a good thing. If you can avoid it, you have to avoid it. Anyway, this is another perspective of how to best increase rejection of cancer in your body, limiting transfusion and minimizing exposure to foreign antigen or things like that. But I don't know how the lymphatic discussion plays a role there, but keep that in your mind as you develop your theory. And then the Tempest data, there's a Tempest data. We can get all the, so Liam, follow up with Tempest where they are, because they decide to help us with that mucinous. And remember you and Eugene O wrote the proposal?
Yeah.
And You know, the next question, how can we get the data? Where did you guys decide to share us the data, and what is the best way to share data? And if we get the data, then Minjung, Eugino, Jisang, Jiyeon, you all can analyze this RNA sequencing data. There's a GEP analysis, gene expression profiling, and then you can see, ask this question, what is unique about this type of cancer that aerogenous spread is more common than lymphatic spread? And you know, we can look at the difference in biology and pathways. So Liam, follow up on with Tempest.
Did they reach out to you?
I don't think I have gotten any email about that.
Oh. Did they mention it in our? I just don't remember hearing anything about it after.
Submitting proposal.
I didn't
Yeah.
hear anything. Just yeah.
Yeah,
Email.
okay. But did they tell you that they approved the submission?
They said they are considering it. I didn't get like positive yes.
Oh, okay. So we'll follow up.
I'm getting few pages. Let's take a break. About 10 to 15 minutes. I have to answer the pagers.
Okay.
Okay. Okay, I answered all my pages. Okay, Jian. Anything else?
Yes, I have more. Let's see. One second. So, this is the format. Would be the format of this manuscript. Did you confirm this one?
Indication, histology, stage. I think stage can be part of the indication.
This is indication.
Yeah. Because this paper is limited to lung cancer.
Mhm.
Oh,
Mhm.
yeah. So double lung transplant for lung cancer, what's the appropriate candidate? And stage is just a part of it, the indication. Yeah.
Okay, all right. Okay.
Yeah.
Then I'm still writing, so I will show you later.
Okay.
It's all written in Korean.
Okay.
And move on the next project. So about the ctDNA in HER2-positive breast cancer, actually I submitted IRB yesterday, but the IRB committee said that it is not allowed for me to be on PI because I'm out physically not in the hospital. So I changed the PI as Dr. Isu Che, and this morning I submitted again. I was pretty shocked. They abandoned me. Fine, okay. And the regular review would be October 16th, but it is already closed. So our IRB can be reviewed at November, I guess. And about the FFPE, actually the practicing and the pathologist in my hospital, they communicated together with me directly. And then we found out that they need both HNA slides and FFPE as well. So we confirmed the minimum tissue volume, and my pathologist is now figure out the cases which are available.
I have a great idea. So H&E, let's scan it in your scanner. 경북대학.
Scanner, what kind of scanner?
So high
Rear
resolution.
scanner?
Yeah, and let's send it to Lunit. And then they can calculate the TIL score and TIL subtype.
Do we need to use a digital scanner? Or a
Yeah,
real
digital
paper
scanner,
scanner? I'm
yeah.
sorry, I don't.
So we can work with Lunit to see, because they're a company. If we say, oh, we want you to come to Chilgo and do scan, the company people will come and scan. But
Is it possible?
That's possible. But if you, I don't know, in 경북 pathology, do they usually scan, or
Not actually.
no? Oh, okay.
They are interested in just pathology, but it is not usual.
Because this is not, I guess this is there's no immunotherapy, but still, we can, this is, trastuzumab, pertuzumab, HER2 targeted therapy, and we do know that TILs play a role in HER2 therapy. Do you
Mhm.
agree?
Oh, yes.
It's not just the targeted therapy alone. We know,
Mhm.
you know, it stimulates the immune cells around. So whether the response rate of just HER2 therapy correlates with the tumor microenvironment, immune microenvironment,
Mhm.
how that plays a role
Mhm.
is in itself a great question.
Sure.
And it doesn't require tissue sample, just H&E slide, JPG file. That's all you need. You don't need a slide.
Oh,
You just need an imaging file,
just the JPEG file?
right?
Oh, okay.
So maybe when you,
Mhm.
maybe do a separate IRB or amend it to
Mhm.
add LUNAT, or just add imaging analysis of the H&E slide. The hypothesis,
Mhm.
maybe just hypothesis is immune microenvironment, immune landscape, the TIL distribution, how that affects the treatment outcome.
Well, actually, I'm working with my colleague, who is a younger professor in pathology.
Mm,
But actually, one of the senior pathologists, she's interested in the similar, but the other project. So I'm not sure she permit this kind of work.
Oh, she has, but you have, but you're gonna send H&E.
I can get the image file from my colleague, my friend. But I'm afraid to ask the senior pathologist.
Mm.
Sorry, she's not friendly. Yeah, so
So maybe present it as just part of this project. This is a one project that looks at ctDNA, but
okay.
we're looking at tissue and we want to look
Mhm.
at the immune cell distribution as the
Okay.
part of the project. And
Okay.
we don't need the slide, we don't need anything but just the scan.
Yes,
Scan,
yes,
scan
okay.
of
I
the
will.
H&E.
I
So
will.
you're not using up any resource.
Mhm.
And pathologists, they don't need to be involved other than retrieve the slide. But if you're making if you need H&E slide for the study, then you know it's the same amount of effort. You just have to do the scan. If the pathology department, you don't want to ask the pathologist, then we can ask LUNAT to come and scan it
Mhm.
at your site. They can bring the scanner and scan
Well,
it.
the problem is that that is not about just the work burden,
Mm,
but if the pathology is involved anyway in this study,
mm,
that will ask me the authorship.
mm.
That is the problem. Sometimes they ask me the corresponding, even they were not do anything in my project. So I wrote so many papers for them. I just shared all of the first authors and the corresponding.
Mm.
But so this is actually a secret of my friend, the pathologist, young pathologist, and me. So if you don't mind, can we just use the JPG file? I'm sorry.
Yeah, just the JPG.
Yes, that was so tough. Yeah, always they call me to their office. Why did you do this? You have to share the corresponding. You have to put my name in the manuscript. Yeah, for 9 years I've been doing.
Oh,
Yeah, still, yes,
oh well.
yeah. Some professors they ask.
Yeah. I mean, yeah. I think first author corresponding is too much because it's only a small part of the project.
Yes, but they do, they ask.
Hmm.
Without that, I will not give you any research about the pathology.
Okay.
So that's why I had to write many papers for me, and the rest of that I had to give them.
Mm.
Yes, just gave them.
Okay, but I'll leave it up to you to how to solve the politics, but.
Okay, yes, I
But
will,
let's,
I
I
will
think it's
figure
a
out the
great
way.
opportunity to get it scanned. So see if you
Mhm.
can get it scanned there. If not, let's call Looney team to come and scan.
Mhm.
And once we, I think we can Liam, we can set up a Lunit teleconference to discuss this project. I want them to be involved and have come and scan if possible, like what this project is and what the hypothesis is, and you know Jian, maybe cite this for this Lunit, right, like half-page proposal. So the whole idea is pill distribution in HER2 positive breast cancer may favorably, may affect the treatment outcome in the neoadjuvant setting. And I think you have an EJM paper or some like HER2 therapy paper that shows that immune cells are important. Not just the ADCC plays a role in HER2 therapy. But in order for them to work, you need pills around. So it's not just the triple negative, but the HER2 breast cancer can also benefit from having a good tumor microenvironment.
Mhm.
So we're gonna apply. We need two things. Number one, Luniscope score. It's called Luniscope score. Number two is
Okay.
the three types: hot, cold, and excluded type. What type is it?
Okay.
So Liam has done it. How to send? He knows how to send the image file to Lunit, but we need to figure out how to scan it in your institution. And I know even they told us that their people will come to Chicago and scan every H&E slide for us. So
Oh,
Lunit is willing to do research. So
the
let's
scan means just the capture or the real scanning.
real scan. Like there's a scanner and you put, yeah,
Oh,
digital scan. It's a digital scanner. Yeah.
yes. Okay.
But high resolution is better for better analysis. So.
Mhm.
Okay, so good. Yeah, I'm glad you mentioned H&E. That's a big plus.
Yeah.
Okay,
I will ask to my pathologist.
yeah.
And since this is almost done, I will show you maybe the next meeting.
Mhm.
And the samples about the metastatic HER2 positive breast cancer, because it should be severe, and actually it is not eligible right now. So I think cohort A with the neoadjuvant setting, the study can be conducted first, and
Okay.
then we'll see. Okay. And about the dividing samples, I studied with my lab members. Actually, the DNA is so stable, so it couldn't be any problem.
Yeah.
But as we can see, there is one paper about describing with the DNA size larger than 100 kb, most sensitive to freeze through degradation.
Mhm.
But for breast cancer, it is only 160 base pair. If even one, it is not even 1 kilo base pair. So
Okay.
I think we can multi it and divide it. That could be no problem.
Okay. Good.
But I'm but I'm still afraid to lose the samples, the wasting samples. What if happen now? Then,
So when you have 6 cc's, 5 to
Mhm.
Predison, Delphi can get
one,
1.
okay,
And then when you do it, you can make a few ones. You know, like if
Uh-huh.
6 cc's make, you know, I would, if I were you, I would do 4. Because you know,
Uh-huh.
we are working with proteomic companies. If they all want, we can always ship their ones to them. We
okay,
have Interven, Biodesics, and then we have Bertis.
uh-huh.
We have Bertis. So 3 proteomic company, we can send 1 cc to all these companies and they can do all different analysis.
But Predison requires at least 5 millimeter.
Predison is 5, yeah.
Yes, then I have only 1 millimeter rust or nothing. This plasma is a 4 to 6. 60.
Yeah, 4 to 6. So it's 4 to 6. So if you, so I mean, it doesn't have to be like strictly 5. I would, Delphi is 1 cc. Did they say, what did Delphi say?
Delphi said 800 microliters, so 1 ml basically.
Hmm.
Okay.
So just give minimum 800 microliter and then
Okay. Uh-huh.
and I would still aliquot like for potential future research, I would aliquot 1 cc, at least 2 1 cc's for
2
future
1 cc.
research. 2 1 cc's for future research.
Okay. Then it could be 2 less for Predison company.
So that's like for, I think marginal is for the Delphi. Well, Interven or Biodesics, they require, don't they require just 100 microliters or something?
So Biodesics asked for 1 cc.
Oh, you did?
And Interven asked for 1 cc. Well,
Okay.
so Interven mentioned 1 cc. You told me that Biodesics and Bertis, since they're proteomics companies, they will only usually require 1 cc.
Yeah, but what did the company say? Because usually they use 100 microliter. I mean, it's okay, let's think about if they are good partners on this project. It's not immunotherapy trial. So it's not really, but then company like Interven can come up with algorithm using glycoprotein to predict how, well, the other thing is, it's not like we have responder or non-responder. Most people did so well. So this is bad training set because so many people did well.
Mm-hmm. Mm-hmm.
But you still have different level of pathologic response. You know, like you have the percentage of viable cells, 0%. And so you do have that and we can correlate with that. So I think 1 cc for future is good. And then Predison should have minimum of 4 cc.
Mm-hmm.
And for Delphi, I don't want to give the whole 1 cc. Let's just ask them because it's such a precious sample. Like, do you need 1 cc, whole 1 cc, or do you need 5 500, microliter, what is the minimum amount you need? Let's just ask them again. So, we want to save up as much as possible. Okay.
There won't be any storage about for the 1cc for future study. So do I need to keep them in my hospital or just ship to the specific place?
Oh, yeah, keep it in your hospital. And then, yeah. Anything else?
That's all I have today.
Okay, good. Jisang, do you have update?
Yes, I want to represent my journal about stats spread through airspaces. Let me share my screen. All right. Can you hear me?
Yeah.
All right. Right. I want to introduce a new concept of tumor spreading, which is called stats, spread through airspaces. I know I want to deal with this topic in terms of mucinous, especially mucinous adenocarcinoma. Right. Oh, I'm sorry. So first, about definition. So in 2015, WHO classification of lung cancer included the concept of stats as a new pattern of invasion in lung adenocarcinoma. So, so it's a different pattern from those that were already known, like hematogenous or lymphatic spread. And this figure on the left explains this concept simply. So one on the lower left, you can see the cluster of tumor cells apart from the original tumor lesion. So the stats is defined as one or more pathologic micropapillary clusters, solid nest, or single cells beyond the edge of tumor into the air spaces in the surrounding lung parenchyma and separation from the main tumor other than tumor islet. So you can see in this figure A, B about micropapillary cluster and CD, solid nest, and EF single cells. Yeah. So the mechanism of stats has not been clearly discovered yet. That's why I said conceptual mechanism. So I gathered many articles, information. So first you can see, I think it's good to see this slide first. So, so as you can see this figure, so stats cancer cells should detach from the primary tumor and overcome anoikis, so which is a form of programmed cell death when they detach from the surrounding extracellular matrix. And third, they should settle down nearby alveoli with co-option mechanism. So first, detach, about detaching. It could be high flow during breathing or mechanical migration by the surgeon's touch when during the surgery, because they could be the causes of detachment, but it's not well discovered yet. And about Anoa'i cases, Anoa'i case, so you can see this figure. So first, there is one article in 2017 revealed the mechanism that how the tumor cells evade Anoa'i kiss. So you can see here normal epithelium. Many of these large extracellular matrix glycoprotein interact with cell surface Anoa'i kiss initiating molecules. For example, fibronectin interacts with integrins. So detachment of these extracellular matrix glycoproteins with this molecule by loss of cell contact makes conformational changes of the molecule and then activate programmed cell deaths. However, in epithelial cancer cells, the MUC1 you can see here. So MUC1, which is very large and heavily glycosylated mucin protein, which is normally expressed in all epithelial cells. They are overexpressed and they lose apical polarization and also interacting with these Anoa'i kiss initiating molecules. So the dense structure of MUC1 protein provides a home-like, sweet home-like microenvironment for the molecule. So it can prevent the cell death mechanism. So it's one of the mechanisms they can evade the cell death
Okay.
during floating. And then I just want to add one thing. So serum KL6 glycoprotein classified as MUC1 is a sensitive marker. So one study observed a positive association between the circulating level of KL6 and subsequent risk of lung cancer. So I think this could be one of available biomarker for evaluation of STAS. And the last, it's settling down with co-option mechanism. So I just found a new article right before. So co-option is kind of here I can say hijacking pre-existing blood vessels of the surrounding non-malignant tissues. So tumor cells exploit the vessels, not making the new blood vessel called as angiogenesis. So one of the article that I found reported the STAS cell were focally attached to the alveolar wall in a manner consistent with the concept known as co-option of the pre-existing blood vessel. So they can use their pre-existing vessel to grow. All right. So that's possible mechanism here about STAS. And then STAS incidence ranges from 14.8 to 60.5% in
That's a lot.
non-small. Yes, quite a lot. So, but the frequency is quite discordant also because variables and interpretation by pathologists are not set up yet. So, and also stats classification also differs all among studies, like the use of distance from main tumor or the number of intervening alveoli, like that. And one recent study showed there is much higher incidence, like 72.3% in the patients with invasive mucinous adenocarcinoma.
Yep.
So before we move on, I want to give a short review of classification of lung adenocarcinoma. So here I just want to point out bronchial alveolar carcinoma, called BAG. So previously it was categorized BAG, but now it's newly classified as AIS and MIA, the lipidic predominant invasive adenocarcinoma, which is non-mucinous type, and also invasive mucinous adenocarcinoma. So I'll let you know.
So go down, go back. So the enteric can be mucinous too, right? No? Look,
It
read about it,
could be.
because it's like a GI type looking, but a lot of enteric.
Right. Actually, these arrows is based on one of the articles I found. So they classified as AIS, MIA, lipidic predominant invasive adenocarcinoma, and then IMA. So yeah, I'm not sure the enteric type is including previously back. So anyway, I want to show these consensus statements in 2021. So it's based on this article. So recent consensus of lung transplantation for patients with thoracic malignancy, it should be limited to very select cases of lung-limited adenocarcinoma in situ, AIS, and minimal invasive adenocarcinoma, or lipidic predominant adenocarcinoma. So actually, this consensus does not mention the IMA type for transplantation.
Yeah, so lipidic, go back to the lipidic. Lipidic predominant is invasive adenocarcinoma, but it's not mucinous. Okay. So I don't know why they don't mention IMA. I think they should. Albert Curie is IMA, right?
Right. I agree. So I think we can add this information
In your review paper, yeah.
Yeah, definitely.
Okay,
Right.
good point.
So and there are some conditions for
Yeah,
transplantation. Yeah, I'll skip this. All right. So let's go back to stats. Stats is related to specific characteristics, like aggressive tumor characteristics, including lymphovascular and pleural invasion, and lymph node and distant metastasis, a higher stage, a high-grade histologic subtype, and also related to the gene mutation. So STAS has correlation with wild type EGFR and KRAS and BRAF mutations. and ROS1 and ALK rearrangement. And male smoking history is controversial, and also based on the result of one article, they analyzed CT imaging, so the absence of GGO pattern and higher percentage of tumor solid component and so lobulated and speculated margin are related to stats. So
Well, this is really
interesting.
important. Like stats was not related to lymph node, was related to lymph node and distant metastasis is very interesting. So because we're proposing there's gotta be a stats without the lymphatic or bloodstream metastasis. A subgroup
Right.
within stats. Yeah, anyway, go ahead.
So I think, yeah, it's what's the cause problem. So anyway, interestingly, the KRAS mutation is quite interesting. So because it's KRAS mutation is the most frequent driver mutations in invasive mucinous adenocarcinoma, about 63 to 90%. And it's also known for association with extracellular mucin and mucin histological patterns. So these results also assume that the stats is related to mucinous feature, I think. And one of the studies has similar results. So I can see here. So the absence of lipidic components, I think, which just mean the non-mucinous type, it's also related to stats. So I think it also support what I mentioned previously. And just I found one study with no correlation between PD-L1 and stats prevalence. All right. So, so a lot of studies prove that stats is an independent prognostic factor for poor recurrence-free survival and overall survival. So there are 3 recent meta-analysis for proving that I mentioned.
So there's nothing about the immunotherapy, correct?
What does that mean about immunotherapy?
These patients are surgical patients. Like, do you have any idea what they were treated with?
So basically, surgery is should be included because the pathology should confirm the stats. But I'm not sure the immunotherapy would be included.
Yeah.
I will look up that.
So, Chitang, this is another project that I would propose.
Mhm.
So, Liam, let's talk about breast cancer project and stats project with Lunit. So Lunit has like 400 or 500 H&E slide coming from Samsung Medical Center. And it has TIL, but it also has information about the tumor islets. So they're able to say stats or not. They have to train them. But I think it's an important feature in lung, whether there is a stats or not. And AI training can also be very helpful, not just say there's a TIL or not, but stats has to be, pathologists call, but if we train, then STAS has a future in terms of predicting response to immunotherapy, and no one has looked at it. So they can easily pay pathologists to say it's STAS or not. Or they can do AI algorithm to say which is STAS. So they can look at our Northwestern sample, they can look at Samsung sample and correlate that with response. So we're gonna talk about it. So when we have a call with Lunit, I want you to present just the basic, why STAS is important, and no one has looked at the effect of it this as a predictor of immunotherapy. But as a oncologist, I know it's a bad predictor. If you have STAS, immunotherapy doesn't work. So no one has proved it yet. So I think Lunit is in good condition to show it because they're a Path AI company. So let's propose. So write like a half-page proposal to Lunit, Qisang, why this would be a good project. STAS recognized by using Path AI and develop that as a future biomarker to predict response to immunotherapy in lung cancer. Lung cancer is predominant therapeutic platform. So let's propose that to Lunit on our next teleconference. Let's schedule that, in a week or two, Liam.
Okay, so you want another one, because,
No, this is same one. So we're going to talk about two projects. Breast.
right, the breast cancer and the STAS.
Yeah.
Okay,
And
yeah, I'll reach out to them.
while we discuss that, we can discuss the third project, which is the what's the other one we said?
Are you talking about the radiomics or?
Yeah, radiomics. Let's talk about that as well. Let's put three
Oh, okay.
agendas. With three agenda in
So-
one call.
The radiomics one, we're planning to set up a meeting in November because their radiomics team is backed up. They were saying they're not ready for a new
Oh,
project
yeah,
yet.
yeah. So November, because their radiomics team has to join. So never mind
Yeah.
that. That's not not in a hurry now.
Yeah, so I'll just send out another email for a separate teleconference in a week or two.
Yeah, so I think he likes 7 to 8 or something.
Yeah, so they said 6 a.m. to 8 a.m., which will be 4 p.m. to 6 p.m. here.
So we'll do 7:30 to 8 on Tuesday.
So that'll be 5:30 p.m. Tuesday for us.
No, it's morning. Oh,
Oh,
you're saying, oh, wait. He said, I got confused. So he likes morning. So it will be, so 6 for them is 4 here, right? So yeah, let's do 4 to 4:30 p.m. so Dr. Chion can present. 4 to 4:30.
4 to 4:30 Chicago time.
Yeah.
Okay,
Either Tuesday or Wednesday.
sounds good.
Okay, Chisang, anything else?
All right. I will keep going. All right. So the interesting point about prognosis is that patient with stage 1A adenocarcinoma and STAS positivity has a similar prognosis to those with stage 1B adenocarcinoma. So I think STAS also might be included in the next edition of TNM staging. I just guess. So,
No, I love
all right. And so I want to talk about the surgery. So some studies showed STAS is a risk factor for recurrence and metastasis in patients with limited resection, not in those treated with lobectomy. And there is one study that noted that when resection margin was greater than 2 centimeter, no local recurrence was observed in this cohort, even in patients with STAS. So I think enough margin can guarantee
Yeah.
no recurrence or metastasis. So this would be suggested for lung transplantation in patients with lung cancer. All right. So, and these two studies demonstrated that separate pulmonary lesions in multifocal or diffuse invasive mucinous adenocarcinoma are very similar and arising from a single tumor and spread in intrapulmonary way. So they revealed this by using molecular profiling and genomic characteristics. Like they compared common gene mutations and proved each tumor lesions has the same mutation.
Good. Yeah, I like it.
So that is another evidence that STAS is the cause of multifocal or diffuse pattern
Yeah.
in invasive mucinous adenocarcinoma.
Yeah.
Right. So this is the final slide. So I made conclusion for all the information. So STAS is related to mucinous characteristics in lung adenocarcinoma and also core feature of how mucinous adenocarcinoma spread. And it also is an independent factor of poor prognosis in patients with lung adenocarcinoma. All
So
right.
let me ask you this question. Number one, is there something similar in GI tumors? I know it's not aerogenous, but in GI tumors, is there something similar that saying that mucinous has with certain feature like STAS is has poor prognosis? Number two, let's say we have GI tumor that has metastasized to lung and they grow. Would that GI tumor, mucinous adeno, can have STAS, or is this a lung cancer unique phenomenon? That's the second question. And the squamous cell carcinoma of lung have STAS, or they never have STAS? So that's my third question. So read about it and let me know what you find. And number four is technical question. How accurate is STAS designation by pathologist? Is there interpathologist variation? Is STAS a well-defined characteristic? I mean, you told us, but, or is it still controversial? Does it vary a lot by pathologist, or is it something that can be quantified easily using AI? So that's like a technical question. Okay, so read about it, and then all these reading can help our discussion during our LUNIT meeting.
All right.
Okay. I'll follow up with Liam after this, and maybe I'll talk to Eileen. But we'll wrap up here for today's meeting, but I'm here all afternoon, so we'll follow up on stuff with Liam and Ivy. Okay. Thank you. It was a great
Thank you,
great lab
Dr.
meeting.
Chen.
Keep reading, guys. This is how we learn. Take care.